Hello to everyone. My name is Darius Kavaliauskas, and I'm going to present guidelines for conducting genetic monitoring in the field for wild cherry. The guidelines was prepared in the frame of the project LIFEGENMON, and those guidelines shortly described wild cherry, its ecology, the reproduction system, and threats. In addition, those guidelines provides the guidance how to select and establish genetic monitoring plot and how to perform field verifiers for genetic monitoring. So as we know, wild cherry is a medium-sized, fast-growing and rather short-lived deciduous tree. And the species is a pioneer and grows in a wide range of habitats. However, the species is extremely scattered, and the distribution is as I said, scattered, and the species is light demanding and is a weak competitor with another tree species. And natural populations are characterized by their small size and occurs in small groups or as a single trees at the edges of the forest or at the gaps at the stand. The species, wild cherry is a species of mixed reproduction system involving asexual reproduction via root suckers and sexual reproduction. The species is monoecious, hermaphrodite, and flowers are insect pollinated. Seeds are spread by birds and by small mammals. And the species sexual reproduction is characterized by a gametophytic self-incompatibility system, which favors outcrossing and prevents self-fertilization. Wild cherry can hybridize with other tree species. So because of this specific biology of wild cherry, it requires additional attention for genetic monitoring plot selection and establishment in comparison to stand-forming tree species. Therefore, because of the Prunus avium hybridization, it is recommended to select and establish genetic monitoring plots at a distance of 8 to 10 kilometers from domesticated cherries. Again, the consultation with local foresters can help to know about the presence, density, and the distribution of forest in the forest of the service tree, wild cherry tree. Orthophotography is very useful because wild cherry can be clearly visible and distinguished from other tree species in the area, especially in flowering time in spring. And of course, initial field survey in the spring when the trees are flowering is very useful, and during the selection trees, and it shows that the trees are reproducing. So again, forest monitoring design for genetic plot monitoring design contains 15 reproducing trees of different genotypes. The distance among the trees should be at least 30 meters, and natural regeneration must be present at the plot. All verifiers, as you can see, the guidelines contains again the list of verifiers and background information, which should be recorded during field work at the genetic monitoring plot. All the verifiers and background information are divided into 3 different levels, to the basic, standard, and advanced genetic monitoring levels. And all those verifiers are described in detail how it should be recorded in a species guidelines. So, for example, within the project, we have prepared a number of illustrations which should be used for recording, for example, bud break or flowering or leaf senescence for wild cherry. So overall, these guidelines provide clear guidance how to perform forest genetic monitoring for wild cherry.